All right, in this video, we'll talk about how the luteinizing hormone and follicle stimulating hormone, what effect those have on male reproduction and what role they play in the menstrual cycle. And then we'll talk a little bit about pregnancy and the role that sex hormones play in that. So FSH and LH are produced by the anterior pituitary, and they act on the gonads in both male and female, although you can tell by the names that they were first identified based on what they do in the female system. So follicle stimulating hormone and luteinizing hormone are named for the effect that they have in the ovary. The sex hormones are steroid hormones, so this is testosterone is the main androgen, and estrogens are, again, testosterone and estrogens are a collection of slightly different molecules, not a single molecule. And then there's also progesterone, and males also have some progesterone. Progesterone, however, is very important in females, especially it's part of the menstrual cycle. We'll talk about that, and also for the maintenance of early pregnancy, critically important. So in the male, follicle stimulating hormone and luteinizing hormone, obviously there's no follicles or corpus luteum have an effect on. So follicle stimulating hormone instead stimulates the Sertoli cells in the testes, which are necessary for spermatogenesis. They help feed and support the growing spermatogonia. And luteinizing hormone stimulates the Leydig cells, which produce testosterone. So a lack of luteinizing hormone will mean low testosterone. A lack of follicle stimulating hormone will mean poor sperm production, low sperm count, and undeveloped sperm. So there is always feedback. Usually it's negative feedback, and that's the case here in the testes and how they interact. And again, you know, coming from the hypothalamus, we have gonadotropin releasing hormone, which is a tropic hormone. A tropic hormone releases another hormone. So gonadotropin releasing hormone acts on the anterior pituitary to stimulate the release of follicle stimulating hormone and luteinizing hormone. So luteinizing hormone is another tropic hormone because it's stimulating the release of testosterone, another hormone. So testosterone feeds back to both the anterior pituitary and the hypothalamus negatively. So as testosterone levels increase, that causes a decrease in gonadotropin releasing hormone and then a decrease in luteinizing hormone. The Sertoli cells produce another hormone whose purpose is to serve as that negative feedback to the anterior pituitary to lower the production of follicle stimulating hormone. So the Sertoli cells produce inhibin in response to follicle stimulating hormone, and that will lower the production of follicle stimulating hormone. So that keeps those levels, this is a homeostatic mechanism. Levels go up, there's a negative feedback loop, levels go back down. For the female reproductive cycle, these hormones produce both the ovarian cycle, which just means what's happening in the ovary as preparation for ovulation and after ovulation. So ovulation is the release of the oocyte. So the maturation of the oocyte, the release of the oocyte is ovulation, and then the formation of the corpus luteum, and then menstruation. That's what's happening in the ovary is the ovarian cycle, and then the menstrual cycle is what's happening in the uterus. So the menstrual cycle is the buildup of the lining of the uterus, and then if there's no pregnancy, the shedding of the lining of the uterus. So that's the menstrual cycle specifically. So during the menstrual cycle, the lining of the uterus builds up. And this is specific for mammals that have a menstrual cycle versus most mammals that have an estrous cycle and not a menstrual cycle. So in an estrous cycle, the lining of the uterus only builds up if there's a pregnancy. So there has to be hormonal signals from the developing embryo in order for that lining to build up. But in a small selection of primates, the apes and us, there's a menstrual cycle. So there's a buildup of this endometrial layer, and then if there's no pregnancy, then the lining is shed. And you can see here myometrium, that's the muscular layer that is not shed. Myo means muscle. And the perimetrium is the outside layer of the uterus. Those are not shed. The endometrium is called a deciduous layer because it can be shed. So within the ovary, the cycle begins with gonadotropin-releasing hormone. That stimulates the release of follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone starts to cause the development of one or more follicles that have it changes an oogonia into a primary oocyte. So the primary oocyte it grows larger, the follicle cells multiply, and they produce hormones as well. And depending on the species, that's going to indicate how many follicles are going to start to develop. So in humans, it's only going to be one. So one follicle, so the ovaries usually alternate cycles. One cycle, one ovary will produce an oocyte. The next cycle, the other ovary will produce an oocyte. If a woman only has one ovary, then that ovary will produce an oocyte every cycle. For mammals that have multiple babies at one time, like dogs and cats, each ovary will have several maturing follicles at one time when they are stimulated by follicle-stimulating hormone. And the follicles make a type of estrogen, estradiol, which is the dominant form of estrogen. So these follicle cells, they start to multiply under the direction of follicle-stimulating hormone, and then they are making hormone as well. They're making estradiol. So estradiol starts to increase then as the follicle gets larger and larger, or if it's a mammal that has litters, then there would be multiple follicles maturing, and this is going to increase the amount of follicle-stimulating hormone and luteinizing hormone. So it's like a little positive feedback loop for a short time, for a little over a week. Increasing estrogen causes follicle-stimulating hormone and luteinizing hormone levels to rise. The follicle gets larger and larger. This is a mature follicle, also known as a Graafian follicle. You might remember after the scientist named Graf, who, what was his first name? It's German, Ludwig or something. And we can see here this big fluid-filled follicle at the surface of the ovary. Very large oocyte. In mammals, most mammals, it's around 100 micrometers in size. It's the largest human cell by diameter. All these follicle cells here are still producing estrogen. The oocyte itself is going to be ovulated with a bunch of follicle cells around it. So these follicle cells are going to stick to it and end up going around it. You can see, you know, scientists like to name things. So this blob of follicle cells around here is called the corona radiata. That sounds very nice, doesn't it? And then around the oocyte is the zona pellucida. So yeah, so we like to name things. This is a less mature follicle. You can see the fluid-filled cavity in there is a little bit smaller. So once ovulation occurs, that big fluid-filled, there'll be a spike of FSH. That fluid-filled follicle will rupture and release that oocyte. Then the follicle is going to become the corpus luteum. and it will secrete progesterone. So for several weeks, if there's no pregnancy, it will secrete corpus luteum, or secrete progesterone, and then it will start to shrivel up if there's no signal from a developing embryo that fertilization and implantation has occurred. So once these are increasing, progesterone and estradiol, this is negative feedback on the hypothalamus and on the anterior pituitary to reduce LH and FSH. So another egg will not start to develop yet while progesterone is being secreted. So if there's a pregnancy, progesterone and estrogen will continue to be high, and that's going to continue to prevent new oocytes from maturing. Although we do know that in very rare instances, twin pregnancies, the babies are a month apart in age because a woman does ovulate a second time, even though she's already pregnant. But that's exceedingly rare. What's supposed to happen is the progesterone is supposed to fully inhibit follicle stimulating hormone so that no new oocytes will mature until progesterone levels go down, which will happen in a couple of weeks if there's no pregnancy. So, all right, so here's hypothalamus again, gonadotropin releasing hormone, just like we have in men, stimulates the anterior pituitary to produce follicle stimulating hormone and luteinizing hormone. Both of these will increase, and then as estrogen starts to be produced by the follicle, they will spike. And this spike in luteinizing hormone and follicle stimulating hormone triggers ovulation. And after ovulation, they decline. And I'll show you in a second what's happening with the rest of the hormones here. So let's look at the inhibitory combination here. So estradiol and progesterone negatively affect the hypothalamus's production of gonadotropin releasing hormone. So as the follicle cells start to produce estrogen, and then after ovulation, progesterone is going to go way up because of the corpus luteum. That's going to inhibit the hypothalamus from producing gonadotropin releasing hormone, and then that's going to inhibit the production of FSH and LH. So no new follicles will start to mature yet. All right. So here's our LH and FSH levels. What's happening in the ovary? So this is the ovarian cycle now. So follicle stimulating hormone and luteinizing hormone. The follicle stimulating hormone is what's signaling one oocyte, in the case of humans, to start to develop. So remember at the beginning here, they're stuck in prophase of meiosis 1. So it will finish meiosis and get stuck, or finish the first division of meiosis rather, and get stuck now in meiosis 2. In metaphase of meiosis 2, it will be ovulated in metaphase of meiosis 2. The follicle then contracts and forms the corpus luteum, which again literally means yellow body in Latin, and it's going to produce progesterone. And that's why it looks yellow, because it's all the cells here, these follicle cells, switch to producing progesterone. And there's so much cholesterol there that it actually looks yellow. And then it takes this about 10 days to 14 days to shrivel up. As it shrivels up, progesterone levels are going to go down. So, and estrogen is going to continue to be produced. The follicle cells here were producing estradiol. They're going to continue to produce estradiol over here. Okay, so let's look at those two hormones. So remember, it's the ovary that's producing these hormones. So estrogen is initially very low. Follicle stimulating hormone increases the size of a follicle. That follicle starts to produce estradiol. Initially, not much progesterone is being produced. Estrogen peaks at the same time ovulation is occurring. That's going to trigger, so ovulation occurs, and now progesterone is going to go up because we have a corpus luteum. Progesterone is going up, estrogen is going back up again. Now, this is the point where if there was a pregnancy, everything would change. So if there is a developing embryo, that means that the embryo is starting to produce hormones of its own, and within days. So ovulation here, I mean, look, like this is like 1 week. So that little developing embryo, which is just a ball of cells at this point at 7 days after fertilization, is going to start producing hormones. And what those hormones do is signal the corpus luteum, don't stop, keep making the hormones. Ping, it's like a little ping, like a little signal. We're here, ping, don't stop. And so if there is a pregnancy, the progesterone will continue to go up, estrogen will continue to go up and finally reach a higher level and continue. And in fact, high progesterone is the biggest reason for all of the really fun symptoms of pregnancy, like nausea and vomiting in the morning, water retention, fatigue, crabbiness. Oh, that's progesterone. That's high progesterone. That's what that's doing. But it's also very necessary to maintain the pregnancy. Women who have trouble maintaining this high progesterone level sometimes have multiple miscarriages and have to take progesterone supplements to keep their progesterone high enough until the placenta takes over making progesterone. And remember, these are feedback inhibitors. If we go back, so progesterone, if there's a pregnancy, progesterone and estrogen are going to continue to get higher. Those are going to feed back negatively on the hypothalamus and on the anterior pituitary, so we don't get any more oocytes maturing. And we'll get no further ovulations if there's a pregnancy. So if there's no fertilized embryo, the corpus luteum starts to disintegrate, progesterone levels drop, estrogen levels drop, because remember the follicle cells here in the corpus luteum were also producing the estrogen. So both of these are going to drop way off, and then that triggers menstruation when they get low enough. So here we have, again, this is the same graph with the hormone levels and what's happening in the uterus, which is the, that's the menstrual cycle or the uterine cycle. So menstrual flow here, loss of the endometrium. The endometrium starts to build up again under the stimulation of estrogen. Builds up, that's the proliferative phase. Ovulation occurs, continues proliferation, and now we're in what's called the secretory phase, which is because the endometrium is secreting nutrients that an embryo, if there is one, could stick in here and get some nutrients to survive while the placenta is forming. Because really, the embryo has to survive about 14 days without very much nutrient at all from the mother. The first 7 days, it's traveling down the fallopian tube and making its way to the endometrium. Then there's another period of 7 days or so where there's really, there's no placenta or attachment, and it's just getting a little bit of nutrient by diffusion from the endometrium, and then the placenta really starts to form, and the embryo is going to really start getting nutrients from the endometrium as the placenta forms about 14 days after fertilization occurs, which would be right when the menstruation would occur if there's no pregnancy. So that's when things really start. But by this point, the little embryo has already been producing its own hormones to maintain the corpus luteum. And the corpus luteum is maintained if there's a pregnancy by the presence of chorionic gonadotropin, is the one they test for with a pregnancy test. That's what they're testing for, is this hormone that the only time this is produced by any human cells is by a human embryo. There's no other cells that produce chorionic gonadotropin. And its job is to maintain the corpus luteum. So, okay, so the ovarian cycle and the uterine cycle are coordinated and affected by the same hormones. The uterine cycle is affected by the hormones that are being produced in the follicle. The ovary is affected by the hormones that are being produced in the anterior pituitary. They don't affect the uterus directly. Follicle stimulating hormone and luteinizing hormone do not affect the uterus directly. The uterus is being affected by the hormones that are being produced by the ovary follicle, estrogen and progesterone. So humans have something weird, which we talked about a little bit in the human evolution chapter, and that's menopause. This has evolved in humans, no other primates, not even chimpanzees, our closest relative. And menopause is a cessation of reproduction, but with still years or decades of life left. This evolved by natural selection. It also evolved in two kinds of whales, orcas and short-finned pilot whales. There could be others that we haven't identified yet. These we have been able to identify because both of these whales are kept in captivity. It would be difficult to determine this in the wild unless, although there have been some orcas where this has been documented in wild orcas that are being very closely tracked for many, many years by specific scientists. And it's been shown in orcas, in this wild orca study that I'm thinking of, that because orcas live in family groups, that in groups where there is a postmenopausal adult female, the babies are more likely to survive. So the babies that are the grandchildren and great-grandchildren of the postmenopausal female, because she knows where the food is, she actually helps catch food and lets the grandchildren and great-grandchildren eat it. So she helps her great-grandchildren survive. And that's thought to have been the same thing that's happened in humans. It's been shown that in hunter-gatherers, if there is a maternal grandmother present in the household, the children weigh more. The children are more likely to survive to adulthood. So that would be something that would definitely be a positive in natural selection. So what's the difference between a menstrual cycle and an estrous cycle? So a menstrual cycle is actually pretty rare. There's a few apes like chimpanzees and gorillas, and a few others, I can't remember, orangutans, that have menstrual cycles, and a couple of other weird animals, a few bats, the elephant shrew for some reason, that have a menstrual cycle. So what a menstrual cycle means is that the endometrium builds up before ovulation and then has to be shed because there's too much of it to reabsorb. In an estrous cycle, the endometrium does not build up before ovulation, and instead, the endometrium only builds up a little bit before ovulation, and then only if there's pregnancy hormones appear, meaning that the embryo is producing hormones, then the endometrium builds up more. So it's less wasteful, right? Because this has been thought to be, having a menstrual cycle is kind of wasteful because you're actually losing blood. So there must be an advantage to that. And this has been a cause of great speculation. What is the advantage of having a menstrual cycle and specifically building up that endometrium before there's a pregnancy? And it turns out there is an advantage to that, is that it kind of gives the mother's body more control over the formation of the placenta. So embryos that are abnormal, so in animals that have a menstrual cycle, embryos that are abnormal are far less likely to be able to implant than in animals that have an estrous cycle. So what that means is that embryos that are abnormal, they have chromosome abnormalities or whatever that are not going to be able to be successful, are going to miscarry extremely early in the pregnancy, and that saves resources. In animals that have estrous cycles, they are more likely to give birth to multiple offspring, where that would be less of an issue. So the primates, besides us and including us that have menstrual cycles, give birth to single offspring, and it's a big deal if you waste a lot of resources in terms of, natural selection. If you waste a lot of resources on an embryo that is destined to miscarry because it has chromosomal abnormalities. There's some other general differences. So in animals that have estrous cycles, they also tend to have a really obvious ovulation. They have external signals to signal males that they are ovulating. And they generally ovulate only a few times a year, sometimes even exactly 1 day a year, depending on the species. And then that's it. And then they're not sexually receptive for any other time other than when they're specifically ovulating. And that's another difference. Animals that have menstrual cycles, including humans, tend to have what's called hidden ovulation, where there's no obvious external signals that they are ovulating. So placental mammals, as we've talked about already in the previous chapter, the embryo develops completely within the mother's uterus and is delivered as a mini adult, and not as an embryo or a fertilized egg. And this has been selected for by natural selection, probably obviously because you can carry around your vulnerable developing embryos instead of leaving them in an egg somewhere where they could be vulnerable to predators. So conception means fertilization. Conception is kind of the medical term, not the biology term. The biology term would be syngamy or fertilization. This generally occurs in the fallopian tube or the oviduct in humans, and the zygote will start to divide immediately by mitosis, and by the time it reaches the uterus a few days later, it's a blastocyst. It's a ball of cells, or a blastula, and looks kind of like this. So this is the outside here is the trophoblast. That's what's going to end up forming the placenta. And then there's a little ball of cells on one side here, the inner mass it's called. That's what's going to actually form the baby. The outside here is going to form the placenta. So here we have ovulation. And the ovulation isn't going to always be, right here by the fallopian tube. The way it's shown here, it can be off to the side here a little bit. But there's a flow of fluid into the fallopian tube because of the action of the cilia that are inside. And so that flow of fluid is going to be drawing that egg down into the uterus. Now, of course, sperm have to swim against the stream here and swim up, and sperm make it all the way to the end of the fallopian tube, although fertilization can happen anywhere along the fallopian tube. If it happens too late, if fertilization happens in the uterus, probably the embryo is not going to be able to implant, because it's going to flow out before it gets to implant. So generally it happens pretty early, and then by the time the embryo makes it to the uterus, it's ready to start trying to implant, because it has to get to this stage before it can implant these. And that's why when they do in vitro fertilization, which in fertilization outside the body, they don't implant the embryos right away. They have to wait until they get to the blastula stage in order to implant them, because otherwise they'll just fall right out before they are ready to implant, because they have to kind of mimic the transit time through the fallopian tube. So once the blastocyst forms, it can implant in the endometrium, and if it's lucky, it will be in the uterus in the right place and be able to implant in the right place. So the medical term for pregnancy is gestation, and that means you're carrying at least one developing embryo in the uterus. Could be 1, if you're a dog, could be 12. Same for pigs, like the pigs that we dissected. They can have 10 or 12 babies easily. So how long pregnancy is correlates with how mature the babies are at birth. So dogs only have a gestation period of 60 days, and their babies are born fairly immature. At birth, both dogs and cats, their eyes are still undeveloped and they're sealed closed. Their ears are sealed closed. Really all they can do is eat and poop and whimper a little bit. When they're born, they can't walk yet or anything. Humans are a little bit more mature than that at birth. Our eyes are open, but we can't see very well. Our ears are open, but we don't hear super well at birth. So we're a little bit more mature. Other animals are born very mature. Like if you've ever seen video of a horse being born or a giraffe, they're born very mature. They can stand up and they can run within hours of being born. And that's fairly typical for herbivores who are prey animals. And human gestation is about approximately 9 months, and there's some debate going on in the scientific community right now about how accurate this 40 weeks business is, because the length of human gestation being 40 weeks was decided by a French doctor in the 1700s, and nobody has ever really questioned it or tested it. And it's probably not exactly 40 weeks. It's probably a little longer than that, and it does vary. It's not exactly 40 weeks. For dogs, it doesn't vary much at all. 60 days means it's like 59 to 61 days. That's their gestation period. For humans, you know, healthy, normal gestation can vary by a few weeks either way. So the implanted embryo starts to make hormones, and one of those hormones we can detect pretty easily, human chorionic gonadotropin, and this is how it's written with a little h, because this is the name of the hormone, chorionic gonadotropin, and then the little h indicates that it's the human version, so that's why that's a lowercase letter for HCG. This is what pregnancy tests test for. The little embryo starts to make this about 1 week after fertilization. So that's before the woman her period is even late. It makes it in extremely small amounts, and we can now detect it almost as soon as it's being made. So often when women are undergoing in vitro fertilization and they want to know very, very quickly if it has worked, they can get a test before their period would have even been due yet, and they can tell. So we can do, and when they're doing scientific studies, generally they're not doing this for normal medical diagnostics, but if you're part of a scientific study about pregnancy, they will start testing for HCG days before the menstrual cycle is even due to end, and it can be detected. Because remember, the presence of these hormones is the reason that the menstrual cycle does not occur. The reason that you stop menstruating when you're pregnant is because HCG maintains the corpus luteum. And the corpus luteum then continues to produce progesterone, and the follicle cells continue to produce estrogen, and in fact those levels will continue to go up as HCG increases. And in fact, it's a quantity-dependent relationship. So the more HCG is present, the higher the levels of testosterone or of progesterone and estrogen. And if there's multiple follicles, it will also be really high. So women who have multiple births that are not identical. So if a woman happens to have fraternal twins, that means she had 2 follicles that matured and she has 2 corpus luteums. And then if she has 2 embryos producing HCG, she's going to get more progesterone and estrogen than a woman who's pregnant with a singleton, and she's going to consequently have worse symptoms, and the symptoms can be really bad if she's pregnant naturally with twins or triplets who are not identical. Now, if they're identical, that means there's only one corpus luteum because they're from one egg. Not all embryos are going to develop, and many spontaneously miscarry. And this is often due to chromosomal abnormalities. And it's been found, interestingly, that the miscarriage rate is way higher than anybody thought now that we can detect HCG much earlier. In fact, it's about 50%. So when they did a study on young, healthy women, so women between 25 and 30 who were trying to get pregnant, and they tested their HCG levels every day during the luteal phase to try to pick up pregnancies really early, they discovered something really surprising, and that was about 50% of the time when they could detect the presence of HCG, it disappeared, meaning that the embryo miscarried really early. So without even any symptoms or any idea that there had been a pregnancy. So, and there again, that could be the advantage of having this very thick endometrial layer, is that embryos that are not able to form a placenta are going to be lost earlier, but they would have inevitably been lost later anyway. If the fertilized egg ends up somewhere else, that's an ectopic pregnancy. And it most often, if it's going to be an ectopic pregnancy, it's going to end up in the fallopian tube, but it can also end up outside in the abdominal cavity. Both of those situations are a medical emergency. If there's a pregnancy in the fallopian tube and it ruptures, it's going to be extremely painful. There's going to be excruciating abdominal pain. And if this ruptures, there's a decent chance that the woman could bleed to death. And so it has to be removed. If that happens, there's no way around it. There's no way to save that pregnancy. You can't put that embryo in the uterus because it's already started to form a placenta with the fallopian tube. And because there's no endometrial layer, that means that it's sticking its little developing fingers, which I'll show you what that looks like as the placenta starts to develop. And as soon as it hits a blood vessel or ruptures the fallopian tube, there's going to be a lot of bleeding. And that's a very serious medical emergency. Luckily, very rare. So during the first few weeks, the endometrium is supplying the nutrients to the embryo, and they're getting into the embryo mostly just by diffusion, but that outer layer, that trophoblast of the embryo, is going to start forming the placenta. And most of the placenta is actually formed from embryonic tissue. So after the baby's born, the placenta comes out, and it's quite large. A human placenta is surprisingly big. It's bigger than a dinner plate and thicker than a dinner plate. I had a professor once in college whose wife had a baby during the semester, and he brought the placenta in to show us in a big black plastic garbage bag and slapped that thing out on the countertop and let us all come and look at it, and it was shockingly large. I still remember that to this day. And this ultimately is going to form an excellent connection between the mother's circulatory system and the baby's circulatory system, where nutrients and gases and wastes are going to be able to cross by diffusion across that space. But the mother's blood and the baby's blood don't actually mix. They're still separated in different blood vessels because, of course, the baby's tissue type and the mother's tissue types are different, and the baby's blood type and the mother's blood type might be different. So here's what a mature placental layer looks like. We have maternal blood circulating inside of the placenta, and the fetal blood vessels that are going to lead into the umbilical cord. So they're actually quite close. The maternal blood and the fetal blood are actually quite close. And the reason that the fetal blood can pick up the oxygen from the maternal blood is because the fetus makes a different kind of hemoglobin. It's a different gene, the fetal globin gene, and it has a higher affinity for oxygen than adult hemoglobin does. So it's able to pick up oxygen from the maternal blood because it has a stronger affinity. And then as soon as the baby's born, the gene production switches, and it switches over to making adult hemoglobin instead of fetal hemoglobin. But most of this is going to be shed after birth. And you can see why this is a bleeding risk for the mother, because the uterus has to really contract after birth in order to clamp down all these blood vessels that get literally ripped away when the placenta is delivered. Now, for multiple births, there are different kinds. Monozygotic or identical twins are natural clones. They are from a single zygote, so a single egg and a single sperm, and so they are genetically identical. Occasionally, monozygotic twins don't completely split. The embryo starts to develop, and generally when it's at a slightly later stage, after implantation for several weeks into development, that's when the split occurs for identical twins. It doesn't occur early during embryonic development. And sometimes they stay together and they don't completely split. And luckily, this is really rare. Conjoined twins are really rare. And identical twins don't run in families, which means it's not genetic, which means it's caused by something environmental. And environmental could mean something in the uterus too. We have no idea what. But identical twins have been around throughout recorded history, so it's not something caused by modern chemicals or anything, although having in vitro fertilization does increase the chances of having identical twins. So something about manipulating the embryos outside of the body increases the chances that later, after they're implanted, they're going to split. The greatest number of identicals ever born, and I just checked this the other day to make sure that it was still true. The greatest number of identicals born are the Dion quintuplets, who were born in 1934, and they were actually sextuplets, but one of them miscarried or died during the pregnancy, and so was not born alive, and was actually like a really small, like shriveled up fetus. They're the only identical quints to ever be born alive. There have been many other claims of identical quints. In fact, in Mexico 10 years ago, there were 5 girls born, but it turned out they weren't all identical, which happens a lot when there's large multiple births, that if they're all the same sex, people think they're all identical, but every time DNA has shown that no, they weren't identical. So identical quints, we have exactly 1. Identical quadruplets are quite rare. 70 sets are known worldwide right now, who are alive right now. Identical triplets are more common than that. Identical triplets are common enough that you may have actually known a set of identical triplets in your life, maybe not. Identical twins are common enough that probably everybody knows a set of identical twins and went to school with a set of identical twins. I've only met 1 set of identical triplets in my entire life, so those are much more rare. Multiples who aren't identical are more common. Dizygotic or fraternal twins, these twins are not any more closely related than any other siblings. They're made by, caused by multiple ovulations. Some women occasionally will have both ovaries will have an oocyte develop and mature, and both ovaries will ovulate on a cycle and form 2 separate embryos. Naturally occurring triplets and quadruplets that are non-identical also occur, although it's very, very rare. Dizygotic twins are pretty common. You probably also know at least one set of dizygotic twins. During my life, have met at least 4 or 5 sets of dizygotic twins who weren't the result of in vitro fertilization. They just occurred spontaneously. So dizygotic twins can be different genders. Identical twins, though, will be the same because they're genetically identical. Now, the opposite of conjoined twins would be dizygotic twins confuse and form what's called a chimera, and you can end up with one normal-looking baby at birth, and chimeras are usually discovered completely accidentally because of genetic testing. We don't have any idea how common it is because usually they're found completely randomly, and this has only even been possible to determine if someone is the result of a fusion of 2 embryos just in the last 20 years or so. Before that, we didn't have DNA testing. It wasn't possible. And now what happens if it's a male and a female embryo? Well, the testosterone that's being produced by the male embryo will dominate, and you'll end up with a phenotypic male embryo. But there are individuals who have been found who have both a testes on one side of their body, an undescended testes, and an ovary on the other side of their body. So it might be more common than we think because we're not looking for this. Some individuals who are intersex might actually be chimeras. Dizygotic twins do run in families. So a woman who is a twin is far more likely to give birth to twins than somebody in the general population. So that tells us that there's a genetic component in the female line, which kind of makes sense because ovulating multiple eggs, that seems like something that would likely be under genetic control. And in fact, it is. So if you're a woman and you are a twin, or your mother is a twin, or your father is a twin, you're much more likely to give birth to twins. The highest multiples that have ever survived are octuplets, and there are several sets of octuplets now that have survived. These are all the result of fertility drugs or in vitro fertilization. There have been several documented pregnancies of 9 or more, and not one of those babies has ever survived. So that is not survivable with current technology. And even with octuplets, there have been many documented pregnancies of 6, 7, and 8 babies as well, where none of the babies survived at all. So anything above twins is considered very high risk, and twins are still considered high risk, especially if it's a first pregnancy. If it's a second pregnancy or a third pregnancy, multiples are more likely to go longer, closer to full term, and be born larger. That's the primary risk with these multiples, is that they're going to be extremely premature. So the first trimester of the pregnancy is the period of what we call organogenesis. So all the embryo goes from being just a ball of cells to having a nervous system and a digestive system, and the kidneys start to form, and the liver forms, and the nervous system forms, and the brain starts to form. So all of those things are in place by the end of the first trimester. In fact, by the end of 8 weeks, most of the major structures are in place, and the embryo switches to being called a fetus at the end of 8 weeks. And it's quite small, only about 1 inch in size, and that's kind of what it looks like. The second trimester, the fetus is mostly growing larger and the brain is developing. The nervous system is developing. The fetus starts to move. Typically, mothers start to feel the baby moving at around the 5th month. They used to have a name for that. In Old English, it used to be called the quickening. I think there was a horror movie called The Quickening a few years ago, but that's what it was called, when you could, and that was considered to be when you were really pregnant, because they didn't have any pregnancy tests back in the olden days. So once the baby was moving and the mother could feel the baby moving, that's when you were considered to be really pregnant. And that was about 5 months. The corpus luteum stops being the primary producer of progesterone by this point, and now the placenta is the primary producer of hormones. This is a little ultrasound here of about a 5-month baby. The third trimester, the fetus continues to grow, the lungs mature, and changes occur that get the baby ready to survive outside without the umbilical cord being there. Labor has to be triggered. Labor is strong, rhythmic uterine contractions that first push the fetus out and then push the placenta out, and then they have to keep contracting to squeeze off all of those blood vessels that have been torn away by the placenta being delivered. Labor is regulated by prostaglandins, which remember we talked about those paracrine hormones. They're involved with several pathways, and including inflammation and pain sensors. And estradiol, and there's oxytocin again, the lactation hormone, is also going to be part of labor. So estrogen being produced over the course of the pregnancy is going to activate oxytocin receptors in the uterus. That's going to help trigger muscle contractions. Eventually, oxytocin levels are going to start to increase. This is going to stimulate uterine contractions. And this can happen. There can be quite a bit of uterine contractions well before labor, kind of practice contractions. And those are the result of oxytocin. Eventually, prostaglandins start to be produced. Prostaglandins are going to have a positive feedback effect and vastly increase the amount of oxytocin. And exactly how this triggering of labor works, why sometimes it doesn't work very well and other times it happens early, really don't understand that yet. This is an area of very heavy research. A lot of research money going into trying to figure out why especially early labor happens, because we don't want babies to be born early. We want babies to wait until they're fully developed to be born, and still not completely understood exactly what is happening and why sometimes this positive feedback loop gets triggered prematurely. Now, if the uterus is ready already, we can give artificial oxytocin called Pitocin to stimulate labor or make labor happen more quickly. Sometimes that works very well, sometimes not so much. Don't know why that happens either. One of the still not completely understood areas of pregnancy as well is how a woman's immune system does not attack the fetus. Because remember, our immune system is trained when we are fetuses to not attack self, and it will attack non-self. But the woman's immune system does not attack the fetus, even though there are a lot of proteins being produced that are not recognized as self by the immune system. Her immune system should attack the fetus, but it doesn't, except in a very few particular situations, like in the case of Rh negative blood and the baby is Rh positive blood. That's a very specific exception. Interestingly, we do scientists suspect that there is some general suppression of the immune system during pregnancy, because women who have autoimmune diseases like rheumatoid arthritis, generally it gets better during pregnancy. And so and a lot of women also report that allergies get better during pregnancy. So that kind of implies that there's some kind of a general suppression of the immune system. To detect disorders before birth, that has improved significantly in the last few years. We have ultrasound imaging, which It can detect many fetal abnormalities. Amniocentesis has been around for what, like 40 years now, where they can remove a little bit of the amniotic fluid, and then you have some fetal cells that you can look at chromosomes and test for specific genetic disorders. We have an easier way now, though. Amniocentesis is not risk-free. It does, stabbing into the uterus does occasionally trigger labor, which isn't good if the mother is only 30 weeks pregnant. About 3% of the time, it causes some kind of a problem. So now DNA technology is good enough that we could just take a blood sample from the mother and test the blood itself for fetal cells and remove them from the blood and test for fetal abnormalities. That way, this is available fairly early in the pregnancy, about 9 to 12 weeks. They can start doing this. So even earlier than the earliest that you can do an amniocentesis. Chorionic villus sampling is you go in vaginally and take a little sample of the placenta. And that can be done a little bit earlier than an amniocentesis, but it's still invasive and can possibly cause an infection or trigger uterine contractions, which we don't want early in a pregnancy. So new diagnostic methods. So we can now do diagnostics for genetic things just by taking a blood sample from the mother. The thing is, though, one of the reasons that people question doing genetic testing is because there's almost nothing you could do about it during a pregnancy. So you could prepare, finding out that your baby is going to have a significant disorder, could help you prepare for that inevitability. And, you know, this is probably something that's going to come in the future. I think it won't be unusual at all 10 years from now for every pregnant woman to get a full genetic test done on her fetus before they're born and know, you know, what their susceptibilities are to all kinds of different genetic disorders, just like you would for an adult. But that has its own problems that I'm sure you can imagine. One of the biggest innovations in the last 50 years is in vitro fertilization to help with infertility. So in vitro is Latin for in glass, and it refers to the fact that we can harvest eggs, and you know, it's way easier to get sperm from the dad, and then you can have fertilization occur in the lab, and then allow the embryos to start to develop, and then pick the healthiest looking embryos and implant them, and that's in vitro fertilization. And this can help when the man has low sperm count. This can help if the fallopian tubes are blocked and the normal travel of the embryo is not possible. Maybe the woman has had an ectopic pregnancy and had to have one of her fallopian tubes removed, and she can no longer have the embryo traveling normally from the ovary to the uterus. There's lots of reasons. Sometimes we just don't know why somebody isn't getting pregnant, and in vitro fertilization is an option that can help. It's very expensive in the United States, but it can work where other things don't work. Sometimes even if there's very few sperm nuclei can be injected directly into the egg and completely circumvent the entire fertilization process, and that sometimes works really well as well. So in this chapter, we talked about asexual reproduction, including budding, fragmentation. So budding would be intentional reproduction, and fission and fragmentation generally not being intentional, but could also cause the production of multiple individuals. Sexual reproduction, which is the general way almost all animals reproduce, produces genetic variation, and that's probably the big advantage. That's why it's been selected for by natural selection, and why it's perpetuated by natural selection, is because having offspring that are genetically variable has been more of an advantage than passing on that individual's exact genetics to the next generation as you would in clonal reproduction. Fertilization originally in animals was external, like it still is today in sponges and cnidarians and many fish and even in amphibians. But internal fertilization allows for greater types of environments to be exploited. You don't have to have a wet environment that sperm can swim in. You can just put them right where the eggs are and provide them with a moist environment internally. Spermatogenesis is the process of producing sperm in the testes, and oogenesis is the process that occurs in the ovary. And we went over the hormones that are involved there: gonadotropin-releasing hormone, luteinizing hormone, and follicle-stimulating hormone. We talked about the ovarian cycle versus the uterine cycle, menstrual cycle versus estrous cycle, what the differences are, and the different sex hormones, and then a little bit about pregnancy. Human chorionic gonadotropin is one of the hormones that's produced by the newly developing embryo that prevents menstruation and triggers the production of a thicker lining and maintains the corpus luteum. And in animals that have an estrous cycle, the production of chorionic gonadotropin will trigger the buildup of that lining and make it ready for forming a good placenta. Okay, that's the end of animal reproduction. There's lots of information in the chapter about human reproduction and birth control and stuff that I didn't want to go over. So if you're interested in that, check it out. It won't be on the test though.